So, very good evening, everyone, for yet another session of MBBT. Today, I hope first of all you're all doing well at your places. Everything is good from academic level, personal level, professional level, physical level, spiritual level, from all the perspective, financial level. There are many levels. So I hope you're all doing well from all perspective, and and will be doing more better and better in your upcoming life. Based on that, so far we have done till our three topics so far. The basics we've done, and then we are done with the cell biology and microscopy, then animal cell culture, and yesterday we have discussed multiple techniques in molecular biology. So we were finished in the 3 days session of that. Now today I will take you to the more subjective part, that is the DNA based markers like RFLP, AFLP, and RAPDs. So these are the topics that we always left out from your studies, but since they are so important, and at the upcoming time in Gujarat, Reliance and many more companies, they're gonna take this onto the industrial level. But these topics are always in the books since the biotechnology has been made, but it was not into the practical level, how we can use them practical level. So it will be more towards the agrobiotechnology side, plant breeding side, but it will be interesting for many students there. And then we will touch a little bit about part of DNA fingerprinting today. So in this DNA fingerprinting, I will be discussing how we can use that for from a forensic point of view. Like if there are some victim there in the crime scene, and we want to look from that DNA, that how if that the victim and the, the samples that we got from the crime scene, is it from the same person? So in that case, the DNA fingerprinting plays a big role. So yeah, that's the two topics that I wanted to cover today with you. Um, so we should not make our time much left out. So far 18 students have joined me. Okay, we have wait for 2 more minutes and then we go ahead, make a kick off to the session. And how's everything? Review writing is going fine. I have to be in touch with these MIT faculty somehow. So maybe I will touch with them today or tomorrow. I just need to grab time and then I will touch with them. So no worries. To give the review of the students. Okay, just 20 seconds more and then we start. Okay. So today, me and my mother, we made one video about masala. Just watch it. So we are just time pass sometimes with the family. Okay. So let's be serious. Not this one. So as I said, these RFLP, AFLPs, restriction fragment length polymorphism, amplified fragment length polymorphism, um, and many SSRs, short-term restriction enzymes, molecular marker assisted screening. So all these markers that we are discussing here, they have multiple use for our life. So like you want to check for drought tolerance trait, you want to check for salt tolerance trait, you want to have higher yield of your plant, you want to be insecticide free one plant. So you want to have many features that you're looking for, but it's hard to have all these features in one plant, in one transgenic crop. So in that case, these markers play a big role. So you can bring multiple traits into your functions, not only in the plants but also in the your dairy animals, cows, sheep, which are helpful for making dairy products. Large production of milk, large production of antibiotics in that milk, having extra nutrients in your milk, some vitamins and minerals in your milk. So all these things are could be managed by these DNA based markers. So that's the appendix. We will discuss about molecular markers and basic principle. Then there are two types of your molecular markers, one based on the DNA hybridization and one on the PCR amplification. Then we go for the RAPD, AFLP, and then for the molecular marker assisted selection. So basically, molecular marker is a DNA sequence in the genome which can be located and identified. So due to some genetic alterations such as mutations, insertions, deletions, the base compositions at a particular location, genome may be different in different plants. So polymorphism can be mapped and identified. So we can plant breeders can detect these genes as we discussed in the molecular marker, but it's very hard to find them. It's not impossible that we cannot find it, but we can find them, and this will make a lot of work easy for many researchers. So these molecular markers are quite highly reliable because they have very many advantages within your plant breeding programs. So you can get to know about the true representation of genetic makeup at the DNA level, and they are quite consistent and not affected by the environmental factors. So molecular markers can be detected much before development of plants occurs, and a large number of markers can be generated as per the needs. Some basic principles of molecular marker here. If there is a DNA marker that can identify two alleles, the genome can be extracted, digested by restriction enzyme, and then separated by gel electrophoresis. So here we have two plants, right? One with a disease sensitive and one with a disease resistance. And we want to check what are the various markers which leads to this particular trait. So what we do is to identify these two alleles. First we get the genome like DNA out of it. Then we cut them, digest them with restriction enzymes, and then separate by the gel electrophoresis. So by this separation, we can check various DNA fragments. So the one with the shorter DNA fragments might be the one with the disease resistance plant, and the one with the sensitive plant might have the longer DNA fragments. So in overall, all these molecular markers of two types. First, as we discussed, DNA hybridization based, that is non-PCR based approaches. And second is your PCR amplification, that is your PCR based approaches. So DNA hybridization and PCR amplification. So let's see this example to identify one molecular marker. So there is a plant A which having a disease sensitive plant, and there is a plant B with a disease resistance plant. So in both cases, we extracted the DNA and digested this with restriction enzyme, and then run over the gel electrophoresis. Yeah, after that is done, so we see on the left hand side shorter DNA fragments, and on the right hand side we see a longer DNA fragments. So both together we can identify from these markers. So these are basically your molecular markers. So this is, if a disease will come, your plant will die and your leaves will tend to die then. But this is resistance plant because it has a longer DNA fragments. So that's the basic principle actually regarding all these DNA markers, to identify them, to screen their genotypes, identify the DNA markers. So based on that, we will do all our studies. In some cases like AFLP amplification, so we will have some PCR based, in some we don't have PCR, in some we have restriction enzyme, in some we don't. So we will, in some cases we have random amplified length polymorphism. So in that way there will be some little bit alteration in different techniques, but overall the basic principle of finding the molecular markers will remain as it is. Okay. So these molecular markers are based on hybridization. So it could be basically your AFLP is included in them because we use also a little bit of radioactivity. So restriction fragment length polymorphism, they are the one with the first technology that were identified for the polymorphism. So they're basically based on the altered restriction enzyme sites as a result of mutations and the combinations of genomic DNA. So there's an outline, we will see how it is done, how we see exactly. Basically in the same RFLP also, basically you have to do isolation of genomic DNA, then you digest it with the restriction enzyme, then you separate by the electrophoresis, and then finally hybridize by the incubating with cloned and labeled microbes probes. So this is the basic outline. You isolate your genomic DNA, then digest it with restriction endonucleases. You will have specific DNA pieces. Then you separate them by the agar gel electrophoresis. Then once it is done, then you transfer over the nylon membrane. Then you incubate with the one that you're looking for, the clone probes, label probes, and then you will see these RFLP bands on it. So, yeah, so that's the basic principle behind it. So here we can see one example, very nice example. There's a plant A and there's a plant B, right? In the plant A, mutation has occurred, right? Leading to the loss of restriction site that can be digested by E. coli 1. So in plant A, whenever we will add E. coli enzyme, it will not digest it anywhere because it has a mutation. Whereas a plant B does not have that mutation, it's a normal plant, it will be digested by the E. coli 1. So hence the result will be, so here we can see in the plant A and plant B, the Hint3 enzyme, both restriction enzymes are able to cut them at the 200 base pairs and leading producing of 800 base pairs in plant A also and 800 base pairs in plant B also. As in the plant B, there is no, the breakage of there is no mutation. So E. coli enzyme could come and attach and it will also cut them into 450 and 300 base pairs. So that's the major difference between your plant A and plant B. So that's why your plant A will not digest E. coli 1, whereas the plant B can digest this. And hence, because of this, when we digest them with these specific restriction enzymes and then run over the gel electrophoresis, we will see different results. First, with the enzyme Hint3 only. So with the Hint3, we will see what we will see: 200 base pair, 800 base pair, 200 base pair, 800 base pairs. So this will be a monomorphic pattern. It will be the same pattern we will see. But as we know in the plant A, we don't have E. coli 1 digestion. So we will have mainly your 1000 base pair. But on the other side, you will have 350 and 650 base pairs of your cutting out. So 350 and 450 plus 250, that will be 650. So here we are seeing a different pattern, that is your polymorphic pattern. So that's why it's called as restriction fragment length polymorphism. Restriction fragment length polymorphism. So this results in the polymorphic pattern of separations at time. I think should this. Let me go. My video is I'm not cut it out the presentation. The presentation is going fine so far. The same thing has been depicted as a cartoon here. The plant source, DNA, and restriction enzymes. Then you do the electrophoretic separation. You can do southern hybridization and then radiography. Then marker based on PCR amplification. The first one that we have talked about, RFLP, that was a non-PCR based. Now we're going to talk about all the DNA markers based on your PCR amplification. So polymerase chain reaction, it's a novel technique for the amplification of selected region of the DNA. So in this PCR based markers, they could be divided into two types. That is, first is locus non-specific and second is locus specific. So first ones are, they can see the locus in that. It is quite like not specific to the locus, where in the second it is very much specific to the locus markers, locus specific markers. So overall, the example in the first is your random amplified polymorphic DNA, that is your RAPDs, and also amplified fragment length polymorphism. That's the advanced version of this random amplified polymorphic DNA, AFLP. And the locus specific markers, they includes simple sequence repeats, SSRs, and SMP, single nucleotide polymorphism. We will now discuss each of these 5 types one by one, and there are some examples also. So random amplified polymorphic DNA, that is the RAPD, they are the random, sorry for that sound, it's mood info. So RAPD is a molecular marker based okay, let's concentrate. So RAPD is a molecular marker based on PCR amplifications, which has been depicted in this figure 53.4. So the DNA isolated from the genome is denatured, and the template molecules are annealed with the primers and amplified by the PCR. Right. So here in this thing, your PCR is playing role. So we are doing the PCR amplification of our molecular markers. The first thing is you isolate your genomic DNA, then you denature that DNA, and then anneal with the DNA template that you want to look for. Then you have a complementary strand synthesis, and then you can amplify that product by the gel electrophorese and can further identify it. The single short oligonucleotide primers can be arbitrarily selected and used for amplifications DNA segments of the genome. So the amplified products are separated on electrophoresis and could be identified. So like in the previous case that we were discussing, the DNA molecules are digesting this enzyme HIND3, but there was no difference in the DNA fragments separated. So however, with E. coli plant A, DNA molecule is not digested, while the plant B, it is digested. So this results in the polymorphic separation. So some applications of RAPD markers, so we can have lots of varieties. Yeah, there's a lot of advantage. So far, a bean variety, Fastulosa vulgaris, which is being produced to identify between morphological traits of different candidates. Yeah. Beyond that, 50 commercial sunflower varieties have been produced based on your RAPD markers. Nonetheless, this produced 296 markers, and that could be scored. So almost 85% similarity were predicted. For example, DNA from plants allows the amplifications of sequences for the A, C, and D, but not B. So A, C, and D fragments we are able to sequence them, but not the B. So this indicates that plant 1 primer sites for the primers might not find the sequence B. So here we can see that that plant A is not able to find a sequence B, whereas the plant 2 cannot find the sequence for A and B. Third, the same, it cannot find the B, but the fourth one could not find the C and D. So we can see that while doing this RAPD markers, so every 4 plants, the 4 varieties, they are showing different trait of your DNA. So based on that trait, so whichever they could find the maximum, we go with that. So RAPD markers have been used to identify several disease resistance genes in plants. So RP94 gene, which is responsible for resistance to stem rust, that is the Piccinia graminis in barley, and RAPD markers identified to link to this gene. So similarly, RAPD markers linked to heat smut resistance, which have been characterized also. Yeah, so some more examples on the same. So there are some demerits also on your, that is, they are constrained about the reproducibility of the results, and non-alleles are also not detected directly in them. Now after that, nucleotide alteration in the genome. So just a second. I need to close my door. So markers based on PCR amplification. So these are the ones, so as we were discussing, which could have genomic DNA from two different plants, often results in different amplification patterns. So these are your random amplified polymorphism DNA. So yeah, so this was random amplified polymorphic DNA. Now comes your amplified fragment length polymorphism, that is your AFLP, amplified fragment length polymorphism. So actually this is a combination of your both RFLP and RFPD that we have discussed. So AFLP, it's based on the principle of generation of DNA fragments using restriction enzymes and oligonucleotide adapters, and their amplifications by PCR. First thing is that you restrict them with the, you digest them with the restriction enzymes, and second is you amplify it with the PCR. So thus this technique combines the usefulness of restriction digestion and PCR. So DNA of genome is extracted. It is subjected to restriction digestions by two enzymes. The rare cutter, that is MSC1, and the frequent cutter EcoRI1. So we cut with these two different enzymes and then ligate them with the oligonucleotides, with the DNA ligase. So this is the mechanism of ALP shown, how the AFLP has been taken care. So you're having this ACCTAGACC, and there is a CATAATTD. So on the corresponding you have a sequence. This is 5' to 3' end, and this is 3' to 5' end. And this is your DNA molecule all in all. So you have your restriction enzymes EcoRI1 and MSC1. So on the left hand side, EcoRI1 tend to cut at 6 DNA molecule, while MSC1 tend to cut a 4 cutter. So it tend to cut 4 from the 4 sequences. Yeah. So this tends to cut your 6, like 1, 2, 3, 4, 5, and then 6. So remaining 4 remaining here and 4 remaining here. And then you ligate them with the linkers. So this blue one that you're seeing might be not in your computer, but these blue ones are the one which is attached with the linkers. So then we do the pre-selective amplification PCR employing single nucleotide extension. So we take the one which has been ligated, which has been with the linker. We will start producing in correspondingly a new sequence. And then once this is done, we will again make it sure that all sequence is done as it is. So all in all, what is happening is we have isolated the plant DNA, then restriction with the we have restriction fragments then, and then we ligate them with adapters. So we ligate with adapters like this U1, and then we add non-specific markers and perform the PCR and then run over the electrophoresis. So all in all, this represents our amplified fragment length polymorphism AFLP, and the lowercase letter represent the sequences found within the amplified region, and the colored lines indicate linkers. Then amplified fragment length polymorphism, that is your AFLP. So AFLP is primarily used in genetic mapping. So several economical important cereal crops such as rice, barley, your wheat have been mapped by the AFLP. So the AFLP markers, which are produced by different combinations of friction enzymes, are distributed throughout the genome. So in barley, AFLP markers are located on the long and short arm of all 7 chromosomes. Beyond that, AFLP markers exhibit strong relationship between number of markers per chromosome and the length of the chromosome also. So more examples here. So in barley map, RFLP compromises around 157 loci, which has been increased to 118 AFLP markers, and polymorphism in the barley have been increased by 12.2 percentage. Yeah, in these different species. Further, AFLP is used for several applications to assist rapid isolation and characterization of target genes. Some merits of these AFLP markers. So AFLP is used to screen pools of plasmid DNA from several clones, and beyond that, it's used to screen large number of polymorphisms. It is possible to saturate specific regions of the genome for map-based cloning of the target gene. And we can apply this analysis of quantitative traits in barley and AFLP also. So, yeah, some on based on PCR amplification. So we have discussed RFLP and AFLP, whereas AFLP was a combination of your RFLP and RAPD. Now let's discuss further in your locus-based markers. So we have discussed the one with the RFLP and AFLP with the known locus based markers, and now we are discussing locus-based markers. That is your sequence tag sites, STS. The sequence tag sites, which represents unique simple copy segments of genomes whose DNA sequences are known, which can be amplified by using PCR. So STS markers are based on polymorphism of simple nucleotide repeats on the genome, and STS have been recently developed in the plants only. So when the STS loci contain simple sequence length polymorphism, that is SSLP, they are highly valuable as molecular markers. And then comes your microsatellites, which are tandemly repeated multiple copies of your mono, di, tri, tetranucleotide motifs. And beyond that, sequence characterized amplified region, that is SARs. So they are the modified forms of STS markers, and they are developed by PCR markers that are made for the ends of RAPD fragments. So SCRs are useful for rapid development of STS markers. So the sequence characterized amplified regions, they are the modified forms of STS markers, and they are developed by PCR markers that are made for the ends of RAPD fragments. So the STS converted RAPD markers are sometimes referred to as SCARs, and they are useful for rapid development of STS markers. Here comes your marker assisted, molecular marker assisted selection, MAS. So they are the selections of desired traits and improvement of crops that has been part of the conventional breeding programs. So the following are the major requirements for the molecular marker in the plant breeding. That is, marker should be closely linked with the desired trait, and marker screening method must be efficient, reproducible, and easy to carry out. The analysis should be also be economical in that case. So we can produce dwarf and semi-dwarf varieties of rice and wheat, and have been responsible for the green revolution. So many developments on the agriculture front are expected in coming years as a result of your molecular breeding. Then comes your linkage analysis. Linkage analysis basically deals with the study to correlate with the link between molecular markers and desired traits. So linkage analysis has to be carried out among the populations of several generations to establish the appropriate linkage. And it's very important your quantitative trait loci, QTLs. So they have many characteristics controlled by the several genes in a complex manner. Some good examples are like growth habit, yield, adaptability, environment, disease resistance, and so on. So, for instance, this golden rice, which is containing this provitamin A, it's involved in the insertion of three genes, and it took them 7 years to do so. It was not an easy process, but it took them 7 years to do so. Yeah. So that's it from the RFLP markers. Though it was very subjective, there was not so much uh, we can do virtual training in them, but it's a must actually for all of you. So that's why I tend to keep these lectures always in this session. So share this. Okay, this is not working. What? It's not working. Okay, I will share them later. It's not working at the moment. So the next part I want to start with you at the moment is, um, oh, there is one assignment. That's good. Provide something back. So yes, DNA fingerprinting. So basically, as we can see some images here, Department of Justice, Federal Bureau of Investigation, Crime Scene Investigation, CSI, that's a famous series. Some very nice fragments, these red fragments of our gel electrophores. This is our DNA, GATC, CGTC, and so on. So all they are predicting that that we are want to make things done in order to find out the truth, to make law and order peacefully. So there are some courses in the forensic science at the moment. If you're interested to do something peace, there is also some cyber security courses that are happening in Delhi Police. So at the moment, the cyber, IT, even though it has provided us a lot of advantage, but nonetheless, people are taking a lot of advantage of this cyber crime and doing a lot of cyber crime, and we don't know how the black market of IT is gonna big in future. So India, we need also some really good, uh, some good places for the same. But these techniques are meant to find out the, in order to find out that 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 the blood sample that we've taken is belonging to that particular species or to particular individual. So DNA sequences are variable and can therefore be used as identifying characteristics. So DNA fingerprinting has advantages over the other sources of evidence. Example, fingerprints, blood type, and etc. And they are quite highly accurate and can be gathered from the trace crime scene evidence. So how we take this DNA fingerprinting? So one way, this is actually your section fragment length polymorphism. The same principle of your RFLP. is placing role. So you digest your DNA with restriction enzymes, cut it into small pieces, and each enzyme will cut a specific DNA sequence. So all human beings at the moment, so out of our 100% of genome, mainly 2% are making the protein. So mainly 2% of genes that are playing role. And within that sequence, due to some there's alteration like 1% alteration of ATCG hormone, these DNA. Because of that alteration in that, we tend to see differences in between our features. Some tend to have some features, some tend to have another feature, and so on. So let's see, there is a BAM H1, and this is our like DNA sequence GGA8TCC. This is in a palindromic sequence that you can read from the left or from the right. It will be always be the same. So your enzyme will come, restriction enzyme. Yeah. So it will come and cut it from there, then cut it from the bottom. And so on. Then you have your sticky enzyme. Then if we say this individual one in which it could cut from the 4 different parts, leading to production of 5 DNA fragments, A, B, C, D, E. And there is an individual 2 which will cut from the 3 different parts, and then we will have A, B, C, D. A, B, and C and D. So we have like individual 1 which have 5 different segments, and then this individual 2 which will have 4 different segments. So let's say there's a crime scene happen, and we tend to find out that the blood group that we saw and the victim is individual 2, and then we wanted to check over the after running over the gel. So we will see 5 on the crime scene and 4 on there, so they are not matching. So we can say that that person is not the culprit, it's someone else. So this way we find out the things. So essentially, once the DNA has been cut by the enzymes, we will have a DNA fragments of various sizes, and each individual bending patterns should have different section enzymes and will cut with each person's DNA at different points, right? So fragments of different sizes will travel different distances along a gel when electric current is passed through it. So this is the different patterns of different individuals, some showing 2 patterns here, some showing these patterns to be separated, then this, and so on. So it's all individuals, like there's 6 individuals, all these 6 individuals showing different pattern here, they are not the same. On the both ends we can see some molecular markers bands. So DNA fingerprinting and let's see how DNA fingerprinting and forensic analysis, they are interrelated with each other. So the first photography of this fingerprint was taken in 1985, and it has been a very nice intersection with the law and science at then. So we can see different unique patterns from one person to the next, and we can use in the paternity disputes as well as in the forensic evidence. So that belongs to my kid or not, if the parents is same or not, so it can help to cover up that dispute also. So we can take the specimens collection could be your licked envelope, dirty laundry, cigarette butt, saliva, anything could be taken, but we have to handle these specimens with gloves, disposable instruments, and avoid talking while sneezing, so that our evidence is not destroyed. So some enemies of these evidences are your sunlight, high temperature, bacteria, moisture. So ideal sample is 1 ml of fresh whole blood, which will have a white blood cells, and we treat with the EDTA, and it's good to go for DNA analysis. So this is the restriction fragment length polymorphism with the individual 1 and individual 2. So you have your the restriction fragment, your restriction enzymes. In the individual 1, they are making 3 fragments, and individual 2, they are making only 2 fragments. So when we separate them, so we are having this A, B, C, D 3 fragments, and we have C and D fragments in this case. So RFLP, yeah, that's how things works. So then we can go for southern blotting to identify the DNA Hybridiz gel. We probe them with a specific DNA, and then we can see the results. So southern blotting we have discussed actually a little bit, but in upcoming lecture I will discuss them in more. So first you run over the gel, then you transfer gel over the nylon membrane, and then you can probe them with the radio label probe that you're looking for, and we can see those sequences with the same sequence. Like if I'm interested with the GCATACC, so we can probe them with them, and we can see that specific membrane will be present there. Yeah, that's it. And there's also variable number tandem repeats, that is your VNTRs, sequence that are repeated multiple times, and number of repeats varies from person to person. So there are 5 repeats and 2 repeats also. So the same sequence GATC is being repeated 5 times or 2 times. So VNTRs usually occur in introns, and VNTRs can be amplified by PCRs and run on agarose gels to produce unique DNA fragments. So yeah, so 5 repeats will be like on this part, and the 2 repeats will be somewhere. PCR we have discussed multiple times. I will not go in that detail. So we have a DNA, you need primers, DNA polymerase, nucleotides, and then you need a thermal cycler. Then the first is the denaturation is happening at 94 degrees Celsius. Then 52 degrees Celsius, the annealing of is done. And at 72 degrees Celsius, elongation is done with this DNA polymerase enzyme. And then this cycle is repeated 30 times with 94, 55, and 72 degrees Celsius, and so on. So you will have, like by the 5th cycle, multiple lot of billions copies. So it also helps application further to identify some, these diseases. You can identify also helps in diagnosing the diseases. So in the normal case, we are having these 376 base pairs, but in the disease case, we are having 2 base pairs after cutting it with restriction enzymes, 175 and 201. So in this way, we can separate between 2 different individuals also, with the normal and the diseased one. We can identify the paternity testing by doing this cell culture, growing them, and then finding that there's a father 1, there's a father 2, and there's a mother. And then after checking this DNA fingerprinting, we see that child has this fragment available, right? So the mother fragments will be there present too, but we can see the father 2 fragments are all similar to the child 1, to the child. So the father 2 is the real father of that child is. So in this way, we can differentiate between different in the paternity testing. On the other hand, in the forensic science, as we have discussed, on the different chromosome level, we can identify it. Right? So is this working? So this is the victim, the sequences of various amino acids, and this is the crime scene. And the suspect has these, so D19, D8, so it seems to be quite similar. So this way we can answer it. That's it, students, from this perspective of DNA fingerprinting. So let's see one example. There's a room on November 1st, so approximately in the evening, 8:15 p.m. So Jimmy Sweet entered his bedroom and looked over his desk and sat over the computer. So while turning his computer on one of the corner, he saw that his shelf is out of the place. So Jimmy shot across the room, closer examination, and he saw that things are ripped off, everything is haphazard. So Jimmy pulled out what happened in the most valuable possession that he has was a Nova lollipop. So that was sticky, and someone was there that tends to lick it away. Yeah, there was some lollipop that he has in his room, and someone has licked that lollipop. So he wants to check who was the person. So he has 7 sisters, Yeah, candy, cookie, sugar, lolly, honey, brandy, caramella. Yeah, so she has these seven sisters, and one of each of these sisters are they love this candy, and they want and we want to find out of these seven who is the suspect who has done this big crime of thefting his candy, the lollipop. So so all these suspects have been detained, and DNA fingerprints of each are now available. So what we have to do is that fortunately the lollipop cannot be licked without leaving behind a bit of saliva. So every lollipop will have these salivas on them. So we tend to take saliva from each culprit here and check it in the lab how things are working. So take it as a joke that what so this is the virtual lab. So first we will add friction enzymes to your DNA sample. So we have this friction enzyme. We add it to our DNA, and then afterwards we will pour agarose gel onto the lab. This is agarose gel here. Then after pouring it, we will put our DNA over this gel. Yes. Now we will turn the power on, and our gel will run over the this. And then we add nylon membrane to transfer these gel uh, particles over the nylon membrane. And then we will add probes. The one that we are looking for to identify them. Now we will place this over the X-ray film, this nylon membrane, so that we can see in the developer and bring it to the developer mode here. So this is the result that we identified at the end. So there's a candy, cookie, sugar, lolly, honey, brandy, caramella. So we take saliva from them and we got the DNA fingerprint out of them. So what we tend to see is that the saliva of the on the DNA, it's this one, and we tend to find out who is the culprit. So answer me in the chat box. So to whom do you think might be the culprit? Is it candy? Is it cookie? Is it sugar? Is it lolly, honey, brandy, and who else? What do you think? Who is the culprit here? Is it honey? Yes, because all these things seems to be quite similar. They are the same DNA fragments. So let's see if the results are fine. So is it? So we tend to put this over this. So this is how. That's why that X-ray films we tend to then put over the things and then check that is it matching? Is it working? I think it's working. She is the one. So congratulations, all of you. You are being recognized as one of the certified forensic lab instructor. So you can join any forensic lab in all over the world, and you can find the suspect now. So you know the technique of DNA fingerprinting, which is very essential in the lab. So yeah, all the best. So we have found honey as a culprit of this crime. So thanks to Nova Lab in this case to solve this. So this is by this nice software has been done. So I can share this software with you also, though it's in the thing that thing. It's still updating. I don't know what's going on here. So I will see. I have to switch it off and then turn it on. So I think that's it for today. So, okay class, so we have discussed basically various DNA based markers today, RFLP, RAPD, AFLP, SSRs, MAS, STRs, microsatellites, and discuss a little bit about their applications, how they are being useful at the moment, and discuss the principle behind them, what is the basic principle is what is working on. And then we have discussed the DNA fingerprinting, what was the basic principle behind it, how it is working, and how can we use them in various applications in our day-to-day crime scenes or any other perspective also for paternity testing and many more. So that's the task for today, today's lecture. Tomorrow we will start with more interesting topic. Tomorrow is a Wednesday, so we will go with protein isolation, quantitation, and purification. This will be with lot of videos, a lot of learning. We will learn from very basic techniques to advanced techniques, your mass spectrometry, right? Your HPLC, chromatography, gas liquid chromatography, and many more. So tomorrow gonna be a long day. I will take a longer lecture, but it will be the interesting one. Might be it go for 2 days or so. So thank you everyone for being there, and see you tomorrow at same time, same place. Wish you all a nice evening. Until then, take care. Stay blessed. Bye bye.